Hi, I'm Francisco Jose Iborra, a co-author of the paper entitled Mitochondria and the Non-Genetic Origins of Cell-to-Cell Variability. More is different. Every single individual in a population has a unique combination of traits. Part of this variability is due to genetic difference, but also to environmental and organism history. In fact, even identical twins show difference in aspect and behavior. However, when we observe a clonal population of cells grown in the same environment, we appreciate difference in size, morphology, or number of cellular components. There is no such a thing as two identical cells in a population of genetically identical cells. This raises a very important question. Which is the origin of this non-genetic variability? The appearance and behavior of one individual, his phenotype, is determined not only by his genetic blueprint, his genotype, but also by how and when his genes are expressed to produce proteins. Two cells in a clonal population always differ in the activity of gene expression, and thus in the number and type of proteins. And these have consequences for developmental and differentiation process, or even processes like resistance of the tumor to chemotherapy. Or even resistance to the virus infection. Gene expression is a process with high energy demands. Around 75% of the cell's energy budget is devoted to produce the necessary proteins. Eukaryotic cells generate most of the energy in mitochondria, which are the cell's power plants that transform nutrients and oxygen very efficiently in ATP. Cells in a clonal population show variability in mitochondrial content, which can have a large impact in the gene expression activity of individual cells. The contribution of mitochondrial content to protein variability is around 50%. This influence is due to the effect of the cell's energy capacity on each of the steps of the gene expression cycle: chromatin remodeling, transcription, and translation. Cells with more mitochondria have higher levels of active histone modification, transcriptional machinery, and activity, which result in more RNA accumulation. More mitochondria imply also higher number of ribosomes and translation activity, enhancing protein production. However, mitochondria do not simply act as a volume knob to amplify gene expression. They influence in an unpredictable way the outcome of gene expression. Eukaryotic cells contain much more different proteins than genes, due to a cut and paste processing mechanism of mRNA known as splicing. Cells with different mitochondrial content can significantly alter the relative level of alternatively spliced mRNA form, changing not only the quantity but also the quality of the proteome. Mitochondria thus emerge as a major modulatory element of gene expression and a determinant of the cell's phenotype. In that sense, dysfunctional mitochondria can result in aberrant gene expression, a relationship well documented in many pathologies like heart pathologies or even neurological pathologies. In a famous article published in Science in 1972 entitled "More is Different," the physicist P. W. Anderson speculated about the existence of emergent phenomena in physics and biology. The total is not equal to the sum of individual parts, and illustrated in this with a dialogue attributed to Scott Fitzgerald and Ernest Hemingway in Paris in the 20s. Fitzgerald said, "The rich are different from us," and Hemingway replied, "Yes, they have more money."